Hey guys, I'm going to pretty quickly go through some of this. I just want to go through it because we're going to be doing a little bit of a project on the genetic disorders. So I just want to kind of run through some of this so that you hear it and you kind of have some foundation for what we're talking about. So has to do with human heredity. So start off by just talking about what a karyotype is. Karyotype shows the complete diploid set of chromosomes grouped together in pairs arranged in order of decreasing size. To find out what makes us human, we have to explore the human genome. A genome is a full set of genetic information that an organism carries in its DNA. A study of any genome starts with chromosomes, the bundles of DNA, and protein found in the nuclei of eukaryotic cells. Scientists then cut out the chromosomes from the photographs and arrange them into a picture known as a karyotype. It shows the complete diploid set of chromosomes grouped together in pairs arranged in an order of decreasing size. A karyotype from a typical human cell, which contains 46 chromosomes, is arranged in 23 pairs. Two of those 46 chromosomes in the human genome are known as the sex chromosomes because they determine an individual's sex. Females have two copies of the X chromosome, while males have one X and one Y. So that's the difference between whether you're male or female. And here's how this looks like on a Punnett square. This is why sex is roughly 50-50. So notice that the female has two X's to give, and the male has an X and a Y. So it is that Y sperm is what determines whether or not you are a male or a female. This just shows you kind of what the X and the Y look like. The X chromosome is much, much bigger. Y chromosome is much smaller. And this actually plays into a whole bunch of different genetic things that are sex-linked, because if you have a trait on this X chromosome from your mom, and then even if it's a recessive trait, if that trait is not on the Y chromosome, then you can have it even though it's recessive because you don't have a dominant trait to mask it. So you get some kind of weird things. And the same thing over on the Y chromosome. This Y chromosome can have things that the X doesn't as well. So this is how we get sex-linked traits. The remaining 44 chromosomes are known as the autosomal chromosomes or autosomes. The complete human genome consists of 46 chromosomes, including 44 autosomes and 2 sex chromosomes. And don't worry about the bottom there. So what patterns of inheritance do human traits follow? The reality, as you're going to see in these slides, is there are a bunch of different patterns. So many human traits follow a pattern of simple dominance, which is what we've talked about so far for the most part. The alleles for many human genes display codominance, which is where two traits show. Because of the X and Y chromosomes determine sex, the genes located on them show a pattern of inheritance called sex-linked. So here is the dominance versus recessive part. This is talking about, for example, a gene known as MC1R helps determine skin and hair color. Some of MC1R's recessive alleles produce red hair. An individual with red hair usually has two sets of recessive alleles inheriting a copy of each parent. Dominant alleles for the MC1R gene help produce darker hair colors. The rhesus factor in the blood, so whether you're positive or negative in your blood, also plays a role in it. For codominance, we talked about this before. There's the ABO blood typing, where A and B are codominant. So if you have IA and IB, then you have AB blood and you're codominant with those. And this is just going through that, but I've already gone through it, so I'm not going to spend much time on that. Genes located on the X and Y chromosome show a pattern of inheritance called sex-linked. Sex-linked gene is a gene located on a sex chromosome. Genes on the Y chromosome are found only in males and are passed directly from father to son. Genes located on the X chromosome are found in both sexes, but the fact that men have just one X chromosome leads to some interesting consequences. This is why most of the different types of color vision and color blindness happen to males a lot more than females. Male pattern baldness is another one that's linked there. If just one X chromosome is enough for cells in males, how does the cell adjust to the extra chromosome in female cells? In female cells, most of the genes in one of the X chromosomes are randomly switched off, forming a dense region in the nucleus known as a bar body. Don't worry about that. Let's see. We're going to skip over some of these. Human pedigrees. So how can pedigrees be used to analyze human inheritance? So the information gained from pedigree analysis makes it possible to determine the nature of genes and alleles associated with inherited human traits. So this is basically what a pedigree looks like. Normally we would do this, but I don't know that we're going to have time. We'll see. We need to get to evolution too. So this is just showing, hey, presence of a white forelock trait, it would be blue and white wouldn't. So this is just showing here are the parents. These three are the offspring because they come directly from these two. These are the individuals that they mated with. And then this group here, this partnership here had 3 kids, and then this 2 had these 2 kids. So this is how human pedigrees work. You can trace traits through there. So thousands of genetic disorders are caused by changes in individual genes. These changes often affect specific proteins associated with important cellular functions. Go through an example. This sickle cell disease, this disorder is caused by a defective allele for beta globin, one of the 2 polypeptides in hemoglobin, the oxygen-carrying protein in red blood cells. The defective polypeptide makes a hemoglobin less soluble, causing hemoglobin molecules to stick together when the blood's oxygen level decreases. Molecules clump into long fibers, forcing cells into a distinctive sickle shape, which gives the genetic disorder its name. So basically, these blood cells end up being the form of like a sickle, like a crescent, rather than being circular, and then they can't carry the oxygen. Cystic fibrosis is another one. Most cases result from the deletion of just 3 bases in the gene for a protein called cystic fibrosis transmembrane conductance regulator, CFTR. As a result, the amino acid phenylalanine is missing from the protein. So you end up with this problem down here on this chromosome, and one of the amino acids is missing. So in this case, this would be the phenylalanine, which would cause this protein to be an odd shape. What this does is, because it's an odd shape, the pump that it's supposed to act in does not work correctly, and that causes a buildup of salt in the lungs, and they get heavy mucus that clogs the lungs and breathing passageways. So it just one little tiny mistake in there causes all sorts of issues. Huntington's disease is another one. So what I want you to get out of this is that one tiny little mistake in the DNA can end up causing a change in the protein, which can end up leading to very, very large consequences and a genetic disorder. Hopefully you kind of see that and understand it. And that's why I'm going to have you guys do a genetic disorder project where you look into one and try to figure out what the problem on the gene level is that causes